Alright, hi guys, welcome to this lecture review. Now this review video have a couple purposes. First is to summarize and highlight some of the key concepts or one of the most important concepts of a given topic. It doesn't present new material, okay? It also doesn't mean that the presented material in this review video is the only thing that is on exam. So, alright, anything listed in the syllabus that is responsible for this given exam, you're still responsible for studying on. Now, warning here, do not watch this video when you're feeling sleepy, okay? I know my voice on video and it is the best non-pharmacological sleeping aid available in this world, okay? It will definitely make you feel sleepy and fall asleep, okay? Watch it when you are alert, alright? Or after a cup of coffee. Alright, without further talking, let's look at some of the most important things about this in this exam, okay? I think the complement system, okay, definitely is one of the most important concepts in this innate immune system. Alright, here is the graph that I took from the original lecture. All right, so there are three main events resulted from this complement system here. First, here we have this opsonizations. The opsonization, in a brief term, really is just to put butters on bacteria. All right, makes it easier for the macrophages to eat. All right, and through this complement system here, we have some other molecule here, the other C3A, C5A being generated. The main purpose to make the vasculature a little bit leaky so that you can recruit more macrophages to the site and do the phagocytosis activity. Another part of this complement system here is to lyse the bacterial cells, okay, and forming membrane attack complex here. Right, so this is the really brief summary of complement system. Now let's look at it individually, okay, in a deeper pathway. Again, this is a picture from the original lecture. All right, so here is the classical pathway here. That's the first to discover, actually third to add. Now this ties to antibodies, okay? We're not going to talk about this here in this innate immune system here. Now first we're going to talk about is this alternate pathway. All right, it is first to act but last to discover. All right, and then we'll also look at briefly the lectin pathway. All right, now ultimately all these things again here lead to proliferations of the pathogen membrane, cell membrane, i.e. forming the MAC, membrane attack complex, and lead to death. All right, and then opsonization, so that make it more tasty, so lead to death again, and recruitment of more immune cells. Here the other term is chemotaxis, okay, chemotaxis. All right, so you get more cells and do all these jobs. So all right, so they all kind of goes hand in hand and ultimately lead to death of a pathogens here. Now to be more precise, these pathogens usually we're referring to bacteria. All right, usually we're referring to bacteria for these innate type of system. All right, let's take another look. Okay, let's take a review side of a way to look at alternative pathway. These are the same slides from the original lecture. All right, to start with the system here, you obviously need to have some C3. Now the first step here is like, out of nowhere you have, not in the blood, okay, a small amount of C3, very small amount. All right, will undergo hydrolysis, okay, hydrolyzed into IC3 here. IC3, what the next step is to bind to this factor B, okay, it's a large factor B, and then here where we have we see our factor D comes in. All right, this factor D does the cleavage, all right, cut down the factor B into two fragments, the bigger pieces and the smaller pieces here. Moving along, this IC3 and BP complex here, actually, in combinations, we refer to as the soluble C3 convertase, i.e. they are in blood, okay, in blood. Soluble means in the blood, okay. Now this entire complex, okay, does the name tells you convertase, convert C3. So it cuts C3, okay. cut C3 into two pieces. Okay, C3A, small pieces. All right, what it does increase vasculature permeability. All right, and then you have your bigger pieces, C3B, which bonds. All right, bonds to the pathogen. So it bonds to it. Okay, covalently. So it's very tight. Stick to it. Now, once it binds to after it binds to the pathogen here, the factor B, alright, factor B comes in again and binds to the C3B. Now, okay, here is the second time we see this factor D. Okay, factor D, what you can remember, D cuts B, alright, it cuts B like the very early steps. Now you're generating a very similar structure than the first time here. Alright, so here it looks like the BB-8 in Star Wars. That's what I think. Okay. Alright, so here this is a structure kind of resemble the earlier the I structure, IC structures, right? So here because of the structure, they're so similar, at least in the presented way, it does very similar job. It has a membrane bound C3 convertase. Okay, now it stick to it, still they're not leaving. Okay, not leaving the pathogen. Okay, this entire complex cut more free floating C3 in the surrounding. All right, chop it up, get more C3A, more further increase vasculature permeability. Okay, you can get more cells into the area, more macrophages. All right, and then the left over here, so you get the bigger pieces. C3B continue to stick on the pathogens covalently, and the concept here is that this whole process will repeat. Okay, basically factor B will comes in again, and then continue. You get more and more on the surface of the pathogen until it's all coated with butter. Okay, and then makes it tasty. Alright, so here is the complement-mediated opsonization. So this concept you may have overlooked. So what happened is that this C3B binds to the surface of a pathogen. Let's try it again. Of the pathogen. So this is the bad guy. Okay, and then bad bacteria, and then it's all coated with C3B now, and then we have our good guy here, which is a macrophage. Okay, or phagocyte. Mainly we're talking about macrophage here. All right, this is a good guy. Now what happened is that, well, you coated it, well, you still need something to interact with this coating so that it take up or eat more easily. Okay, so there are complement receptors, okay, on these macrophages. Okay, complement receptors, CR1 receptors, and these C3B will bind to these complement receptors and right, basically interacted very well and eat up the whole bacteria that way. All right, so another key point about the complement system, like I mentioned earlier, forming as MAC, membrane attack complex. Now this is a simplified graph here, skipped a lot of steps here, but the idea is actually not so bad. Okay, you have your C5, okay, being cut. Okay, being cut, really what happen is the C5 convertase. All right, it cuts into, again, a bigger pieces and a smaller pieces. This is the big piece, this is the small piece. All right, the small piece interact with C6. All right, and then it forms a binding site where the C7 can go, and basically here we have our pathogen. Oops. Alright, and all these complex here, and basically it's forming an anchoring point, and you have 5, 6, 7, 8 already assembled, and your 9 C9 it goes in, and forming more and more and more of this 9 C9 protein, and eventually you are forming a hole. Alright, this is the bilayer, okay, and where waters and go in and go out, and your pathogens will die. Okay. Alright, so another part of the complement system other than activations is its regulations. Now here it's basically the same slides from the original lecture, highlighting some of the key points here. Now again, this complement system is via a positive feedback loop. Okay, if you don't regulate this complement system, it will go on and on and on and on. Okay, now it got too much. The main thing is it's gonna deplete your C3 in your blood. Now anything too much is not good in my idea. All right, so there are several things that you can in place to regulate the system. First of all is this factor P, okay, properdin, okay. I would use this term on exam, factor B, alright, factor P, I mean. So this factor P, like this picture shows you here, it goes into the C3 convertase and stabilizes the structure, so it makes it more regulated. Okay, so it doesn't, it's more in a regulated factions to generate more C3b rather than just keep going and going and going and without a checking. Alright, so you can, when it gets too much on the microbial surface, it can actually, basically degrade some of those. Now in terms of degrading on a microbe surface, this could be including the factor I and factor H. Now these two things need to work hands in hand together. So factor H need to bind to C3b, and where the factor I, okay, comes in to cut it, and basically inactivating the C3b. So I C3b maybe this I is inactive, okay, means inactive. Alright, so here is the catch here. It binds to a sialic acid part on some of the surface of the pathogens, and what happened is that some of the bacteria can have more sialic acid group to fool the system, so make them think, hey, this may be a human cell, or there's some regulator here, telling it, so it degrades more of these C3b, makes this basically inactivating this complement system. Well, in terms of human cell, now looking at the human cell side, we don't want the complement system formed on our cell. So there are, couple factors here. First is the decaying accelerating factor, or DAF. Alright, so DAF is expressed on all human cell surface. Okay, this is on human cell surface. Notice that. Alright, some of the factor kind of free floating. This one is on human cell surface. So its job basically is disrupting this convertase activity here. Kick out the factor BB. Alright, kick out the factor BB, and so you don't have the complex in place, and you don't lead down, it doesn't lead to the activation of the complement system. And then you have another factor here, the membrane cofactor protein, or MCP. Again, this MCP is on human cell surface. Okay, on human cell surface. Now it works kind of like a factor H. It goes into interacting with the C3 convertase, and and then after the interaction, you kick off the BP, and in this case it exposes some site, okay, it's susceptible to factor I, okay, to come in, cut it. Alright, basically you chop the C3b into inactivated pieces, and then you basically stopped the activations there. Alright, so fast forward, and I think other factors that are important in this presented material is the toll-like receptors and its co-receptor here. Alright, I always emphasizing the toll-like receptors mainly participate in recognitions. Alright, so here is the toll-like receptor graphical representation anchoring on macrophages. Alright, on macrophages, and here is an example where there is this lipopolysaccharides on a bacterium being present. Alright, so the receptors on the macrophages that is actually binding to LPS is the CD14, alright, LPS receptor. Alright, and but it binding it also interacts with some kind of a associated protein, alright, so MD2 here. So this graph kind of really shows you, hey, this LPS does not directly binding to this toll-like receptor 4. Alright. Okay, so other cofactor can also help with this presenting or assisting in recognitions. Alright, so when you have binding events or recognition events between this toll-like receptors and its co-receptors here, what it leads down to the end of the end story here is to activations of NF-kappa B, alright, NF-kappa B, and lead to inflammatory synthesis of inflammatory cytokines. So you get inflammations there. Alright, inflammations, and basically you lead to different type of symptoms. Alright, red, okay, redness. Alright, warm, okay, and all these warm to touch, edema, okay, so sorry for that word here. Alright, warm and edemas, and okay, and increase vasculature permeations as well. Alright, so that is the end result. Now, inside there's a lot of complex signaling cascades. Basically, these are all kinase cascade. You're not responsible for knowing these detail. They're basically performing a series of phosphorylation reactions and lead to that activations there. Okay, just remember, sensing of toll-like receptor, sensing of LPS by toll-like receptor lead to activations of NF-kappa B and synthesizing inflammatory cytokines. Alright, so there are other products being released by this macrophages here. So macrophage doesn't only eat up the bacteria, okay, it also does some kind of a downstream signaling event, okay, sending out messenger message to the different systems. Okay, one of the more relevant one is the IL-6. Okay, IL-6, it will lead to fever, okay, lead to fever that we learned about in an earlier course, and also induce acute phase protein productions by hepatocytes, or in other term, it's in the liver. Alright, I always draw a triangle for liver. Alright, and other factors, they are all important. I won't read about directly on the slide, but that's something you don't want to miss. Alright, TNF alpha, IL-1B or 1 beta, CXCL8, and interleukin 12. Okay, all these are very important things to be remembered for this particular topic. Alright, notice I say topic, not saying exam. Okay. Alright, so you have your IL-6 being released by activated macrophage. Here, activated macrophage basically is like, well, once it tastes the bacteria for the first time, activated, alright, once it eat up one. Alright, so like I said, contribute to fever and also leading to release of acute phase proteins. Now, these are the examples of these acute phase proteins. Here, reactive protein, which has role for opsonizations. Now, I think this is pretty straightforward. I won't go into in the review again. And we have fibrinogen leading to clotting and Mannose binding lectins. Okay. Alright, let's talk about the Mannose binding lectin here. And remember that is the second pathway that is leading to activations of the complement systems. Okay. Now here, basically it's, in a very similar way you can understand it. Now here, if first formed by this lectin bind to Mannose on pathogen surface, and here that is leading to activation of this MBL associated serine protease. So it's kind of long name, and MASP is enough. Now this MASP would cut C4, okay, C4, that is free floating soluble C4, again into a big piece and a small piece here, and C4b here is the big piece, which is covalently bind to the pathogen surfaces. Alright, so MASP also cut the C2. Here is a little bit different than the alternative pathway, so it does two jobs simultaneously basically. And you again, once you cut it, always have a bigger piece and smaller piece, and the bigger piece here interact with the C4b. After this interactions here, you get a similar shape here. I always try to be consistent with the shape. In the book that I'm referencing, you have a long piece, and a kind of semicircle or oval type of a shape there. This type of a shape usually represents some kind of a convertase, and here we have another C3 convertase. So we have a soluble C3 convertase, we have a membrane bound C3 convertase, and we have a C3 convertase. Now unfortunately these names are very confusing, but the job is the same. C3 convertase, it converts C3 into activated C3. Now here the activations meaning by cutting it into two small pieces. Alright, so we have again C3a and C3b, and down the road lead to more C3b on the surface of the pathogens. So the initiation part is a little bit different than the alternate pathway, but the end result is always the same. Alright, so here that's the basic summary of the key concept, or perhaps I think the most difficult concept, complex concept. Nothing is very difficult, but it's complex concept in this given exam coming up. So let me know if you have any other questions, and I would end my video here. Well, hopefully you watch it all the way to the end, and hopefully that is a little bit helpful for you to go over the material again. Thank you, and good luck studying.